Okay, we are now going to talk about the extracellular matrix. So extracellular means outside of the cell, and matrix implies this complex kind of network of different types of molecules. Lots of different types of extracellular matrix depending upon the different types of tissue. So lots of different types of connective tissue that is basically a buffer between organs and bones and other organs and things like that. We are talking about this very generally. If you take anatomy, you will learn more about the specific types of extracellular matrix in different types of connective tissue. The importance of the extracellular matrix is it's a way that the cell can connect to the environment. And there are three general types of molecules. The first type is for structure, so strength and flexibility. And these proteins, the characteristic ones that we will talk about are collagen, which is right here, this big purple mass, and elastin. And I think one of the easiest ways to think about collagen and elastin is to look at old people. I'm starting to become an old person. And I noticed that when I look at my hands, the skin isn't as tight anymore. Right? So you've probably heard of people getting collagen implants, right? Puff up your lips, these collagen fillers. Well, collagen will break down over time. Elastin, proteins will break down over time. And what happens is we stop filling the space that used to be taken up by the collagen and elastin. And if you think about your hands, and people as they get older, their hands get wrinkly, right? So we still have all that skin, but now it's not so full of this extracellular matrix and proteins. We also have less strength in our skin. So people, as they get older, their skin is more fragile, right? So it can tear easier, bruise easier, but really it's the tearing, and you can get less flexibility in your joints as you get older because we don't produce all of that extracellular matrix anymore. It gets broken down. Okay, the second type, let's see if I can get rid of this. Whoops. Drawing and erasing. Second type of general molecules found in the extracellular matrix are proteoglycans. So this means proteins and sugars. And proteoglycans are primarily proteins with glycosaminoglycans attached, and that's going to be in our excuse me, next slide or two. We'll talk about glycosaminoglycans. But these are, again, proteins and sugar chains. There's also a third type that's very similar called glycoproteins. And in fact, proteoglycans are a subtype of glycoproteins. This is, again, sugars on proteins. And the ones that we will talk the most about are fibronectin and laminins. So this is just showing you again plasma membrane, we have our cytoskeleton, and then the stuff outside. And it's going to be attached through transmembrane proteins and through proteins with sugar modifications, like we learned about, that happen in the ER and Golgi. And then you can see it produces this gel-forming polysaccharide, and polysaccharide means many sugars, right? So we talked about before, when you get sugars wet, they become sticky. So the extracellular matrix is this nice, sticky, kind of cushiony buffer between some of our organs and bones and cartilage and all that kind of stuff. A type of cell that they talk about a lot in cell biology is fibroblasts. And fibroblasts shown here, and so you know that they are an abundant cell type, and we do lots of cytoskeleton staining of them because they're easy to grow up in cell culture, easy to isolate. What's important for you to know about fibroblasts is they are a type of cell that produces, or more accurately secretes, components of the extracellular matrix. Okay, so they're there secreting all this stuff to make the extracellular matrix, such as collagen, which we talked about, and elastin, those two proteins that are important for structure. Hyaluronic acid is a type of glycosaminoglycan. So they produce other things like enzymes and growth factors, but they're a main component in connective tissue that produces this extracellular matrix. Okay, glycosaminoglycans, GAGs. Why it does that? Okay, basically disaccharides, which means two sugars, shown here. All different kinds. Your book talks about some other times, but they're repeating. So it's disaccharide, 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 like you can see here. So they're these repeating units. The reason they're called aminoglycans is you can see the NH groups. Remember, it's just like on an amino acid, NH groups. So it's a sugar with an amino functional unit. And glycosaminoglycans, as I said before, are the components of proteoglycans. So you can have this big long core protein, that's the proteo part, and then all these sugars attached on it, that's the glycan part. So this is a zoom in. Here's a serine amino acid that has this repetitive structure right here of glycosaminoglycans. So if you look back here in your extracellular matrix, you're going to start to see these long proteins with all these branched chains of sugars. Okay. Proteoglycans. Oh, and I had a picture right here. Again, proteins, carbohydrates, more polysaccharides. So just remember that the extracellular matrix is sticky. Proteins and sugars, that's the main components. And the whole point is to hold things together and give certain cells space to kind of settle down. So depending on the type of connective tissue it is. The next type of protein we're going to talk about is this little guy right here, fibronectin. And what I want you to remember is this interaction between fibronectin in the extracellular matrix and integrins, which are transmembrane proteins. So here's a connection between the plasma membrane and the extracellular matrix. Fibronectins are secreted by lots of different types of cells. Okay? So they secrete this protein that's made up of two polypeptides. So here's a protein chain, here's a protein chain. They're held together by disulfide bonds. And what's unique about fibronectin is you see it has all these things called domains. Domains of a protein just mean regions. And they're regions with a specific characteristic. You can see that fibronectin can bind collagen and heparin and fibrin and cell surface receptors and heparin again. And this RGD sequence stands for the 3 amino acids, arginine, glycine, and what is it? Glutamine? Glutamic acid? Oh, I should know that. Okay, you make sure you look it up. Well, actually, you don't have to know it. RGD sequence. RGD sequences are really important because they interact with integrins. So integrin is that protein that integrates the cell with the extracellular matrix. It's a transmembrane protein. It's important in signaling. We'll talk about that in a little bit, and we'll talk about that in the next unit. But the point of fibronectin is it's an adhesive glycoprotein that helps the cell interact with the extracellular matrix. And what's interesting about fibronectin is, like I said, cells secrete it. Okay? So it can be soluble. So it can be out there in the extracellular matrix, and then it can also interact with integrins. Okay? So these are those transmembrane proteins, and starts to unfold and starts to form this matrix. And now, as you unwind it, and remember it has all these different domains, you unwind it, these different domains can act with other parts of the extracellular matrix. So the cell produces it, it interacts with it, and it starts to build this extracellular matrix. And again, you see this term signaling. Integrins are made up of alpha and beta heterodimer. That's why it's drawn like that. And they're important in telling the cell, hey, this is what's going on outside. This is what you need to do inside. What's interesting is many cancer cells don't make fibronectin. So if you don't make fibronectin and you don't have this extracellular matrix interaction, then you're not going to be bound to a specific location. And the big scary part about cancer is when it metastasizes or when it moves to another location. And so we've talked about, or you're learning about in this chapter, different ways that cancer cells can basically let go of where they're supposed to be. And one method is by not making fibronectin. We talked about cell-cell adhesion or cell-cell junctions in the previous lecture. So cancer cells can stop making coherins, so they stop interacting with cells and tissues. Matrix metaloproteinases. So remember, whenever you see ACEs, that means an enzyme. This is an enzyme that degrades proteins. The metallo part means it needs metal ions to work, and it works on the matrix. So cells can secrete these little proteins here, and it starts, in this case, chopping up the fibronectin. And when you start chopping up the fibronectin, you start releasing yourself from that extracellular matrix. Now, some cells need to do this normally. You may need to be replaced. You may, during embryogenesis, need to move to another location. But again, what's interesting is cancer cells can produce these proteinases and break free from the extracellular matrix and move. So cancer cells have lots of different strategies to move from one location in the body to another. Another component, another glycoprotein, so laminins are adhesive glycoproteins that are part of the extracellular matrix. Laminins are in a particular area. They're down here in the basal part of a cell. So remember last lecture we talked about there's apical, top, and basal, bottom. So lamina, this part right here, is made of laminins. Laminins have this characteristic kind of cross-like structure. And they're kind of a middleman connecting the epithelial layer with the connective tissue. Right? So they act as a barrier so these two cell types don't mix, or these two tissue types don't mix, and they help anchor the epithelium to the connective tissue below it. Just an FYI, and I don't mean to do this to confuse you, but I've got a little confused. Remember back last unit we learned about lamins. That's my problem, I call them laminins. Lamins in the nucleus. And those are the proteins that make up kind of the nuclear cytoskeleton. Okay, these are laminins, and so these are different. These are glycoproteins in the extracellular matrix. Integrins. Integrins are transmembrane proteins. They're made up of an alpha and beta heterodimer. So we talked about heterophilic interactions. Heterodimer means two things that are different, right? So you have alpha, beta. So here's an integrin receptor. They have a couple different properties. They help integrate the cytoskeleton. See up here, you've got actin with the extracellular matrix. They're important in cell signaling, and cell signaling is a type of communication. That's what we're going to learn about next unit. And integrins recognize that RGD sequence on different types of molecules. So we talked about fibronectin. The other thing to look at with integrins is I have mentioned to you that you need to know about linker proteins. Right? So, for instance, integrins at focal adhesions interact with alpha-actinin and talin and vinculin. Okay, we're going to talk about FAKs in just a minute. Integrins and hemidesmosomes interact with plaquins and plectin. So remember that a lot of times there's these linker proteins, these intermediates between, for instance, a transmembrane protein and the cytoskeleton. Focal adhesions are a type of adhesion that relies on integrins. Integrins, sorry. Okay. So here in red is our integrins. Okay. This black line is our plasma membrane. This cell is moving. So focal adhesions are transient, means they come and go. Connections to the surface. Okay, so if you're a cell that's migrating, this might be to endothelial cells, wherever you're moving from. And these are these local interactions. And we talked about lamellipodia. So lamellipodia make focal adhesions. So you've got your actin, you've got your linker proteins, talin, vinculin. You've got your extracellular matrix here in yellow, And so you'll form these focal adhesions. It will let the cell grasp onto the surface, and then you'll break them down, and you'll make new ones in the front, and it allows the cell to move. So focal adhesions are clusters of integrins. Okay, they interact with bundles of actin. They help in movement. They attach and detach. They're being made and broken down when a cell needs to move, crawl. Focal adhesion kinase. Okay, kinase. ACE means enzyme. Kinase is an enzyme that phosphorylates, which means it adds a phosphate group to other molecules. This is going to be super important in cell signaling. So here you have in red and blue your integrins. And here are your linker proteins, and here's your actin, and here you have focal adhesion kinase. And this is important in the cell signaling or the cell communication. And what happens is the integrins receive messages, chemical messages from the extracellular matrix, from this outside environment, and they pass it on to the focal adhesion kinase, which can then phosphorylate some of these proteins. Phosphorylation a lot of times activates, but sometimes deactivates proteins. Again, we'll get to this next week. And it tells the cell what to do. Okay, so it may say, stay here, we're good, let's grow. Move the actin, let's crawl. And again, focal adhesion kinases are important in cancer. So what happens in cancer is the focal adhesion kinase no longer listens to cell receptors, such as integrins, or what we will get into next unit, epidermal growth factor receptor. Right? So we have these transmembrane proteins on the plasma membrane that communicate with proteins inside the cell. These proteins communicate with other proteins, which communicate with other proteins. So it's like a domino effect. And, excuse me, your book talked about this epithelial to mesenchymal transition, and it helps things migrate, especially in embryonic development. But what happens in cancer is FAK says, I don't care what you say, I am going to tell my cell to move, to grow, to survive, to invade. Okay? You might be saying, hey, stop, we're good. I'm not listening anymore. So the cell can move on, it can grow, it can divide, it can cause cancer. Okay, we have just a couple more types of interactions with the extracellular matrix. Hemidesmosomes are a type of cell. Oh, that's what I should erase. It's not a cell-cell junction. Okay, sorry about that. It's a cell junction to the extracellular matrix. So I will fix that before I publish your slides. All right, remember we talked about an apical side and a basement or basal side. So a hemidesmosome is half a desmosome. So just like desmosomes, it has cadherin, well in this case it's showing integrins. Okay, so ignore me. It has integrin transmembrane proteins. It has plectin, and they're also called plaquins as the linker proteins, and the hemidesmosomes, like the desmosomes, are connected to intermediate filaments. And then here's your extracellular. So if you have a cell with an apical side and a basolateral side, its little spot welds are called hemidesmosomes because they're half of one. But it has the same kind of idea. It has a plaque, right? So it's high concentration of interactions with the extracellular matrix in this case, with the intermediate filaments, and then all your integrin transmembrane proteins. This is what gives your skin mechanical strength as well, and allows it to stretch and move without breaking and tearing so much. All right. So it's interesting that I put this here. I split up the talk, obviously, so this is all talked about in the first chapter 15 lecture, the tight junctions, the adherents, the desmosomes, focal adhesions, hemidesmosomes. So look at the different types of cytoskeleton that they interact with, actin versus intermediate filaments, cell to cell, cell to substrate or extracellular matrix, know their different functions. Now this is kind of a weird organization here I'm finding out. Be able to recognize them. There will be some pictures on your quiz with information allowing you to recognize the different types of cell-cell junctions or cell extracellular matrix junctions. Last one we're going to talk about is costameres. Costameres are specific to muscle cells. They are actually attaching the muscle fiber to the plasma membrane, so it's very internal. Okay. You can see that they line up on the Z discs. They also can then reach out beyond that to extracellular matrix, so it keeps muscles intact and where they should be. The important protein that costameres interact with is this protein called dystrophin. And when you have a mutation in dystrophin, you get a weakness in your muscles. So they're not attaching, the fibers are not attaching to the plasma membrane very well. You get muscle weakness, eventually, unfortunately, muscle failure, and this is called muscular dystrophy, because this dystrophin protein is no longer working correctly. So your muscle fibers are not attaching to the plasma membrane, which isn't making a strong interaction with the extracellular matrix. Okay. Again, this should have been in the last, lecture, sorry. But I thought of this as I was writing the quiz. I highly suggest you make a table like this. You got your junction types, write down your transmembrane proteins, write down your linker proteins, and in parentheses I've said how many you need to know. So these would be like for adherent junctions. Your transmembrane protein would be cadherins, and these would be the different kinds of catenins. Okay, and they are linked to actin. So fill in this chart, have it with you for the quiz and the unit exam. I hope this little lecture has helped you connect some of these concepts, and I will talk to you later.